Hello everyone. Hope all of you are doing well. Today we begin with our first class this session on the first and most important thing in biology, that is cell. Let us write it down first on the board. Cell, its structure, function and organization are such topics which keep on coming in various exams, and we will be discussing on various important points in this class which will be quite important for both board exams, competitive exams, whenever and wherever you get something related to cell. Today's discussion, I hope, will be useful for you. So to begin with, cell was first discovered by a person, rather by one of the greatest biologists, Sir Robert Hooke. Robert Hooke in 1665, through his self-made microscope, discovered cell in a piece of cork. Now, let me tell you, cork by itself is a dead tissue. So, what Robert Hooke saw inside the cork was dead cell, because cork itself is made of dead tissue. But whatsoever the case, he was the first person to have a microscopic view of cell. Now, after Robert Hooke, several scientists and researchers did their own job and contributed more to the understanding of cell. First, let me tell you that from this section, who discovered cell, the question is there in various exams. Robert Hooke in 1665 discovered cell, whether it be dead or living. Our point of interest is of course cell. So cell was discovered by Robert Hooke. But later on, sounds like a story, isn't it? But this story includes all our points of interest. So later on, Anton von Leeuwenhoek discovered living cell. Who? Anton von Leeuwenhoek. He used his specialized lenses and he made a handheld microscope, very much like the ones that are given in the biology book these days. It looks more like a selfie stick attached to a mobile phone. It was a long stick, and with that long stick, the main piece was attached, the main piece of the microscope was attached, and with that, he saw a drop of cold water. And he made certain drawings. He saw particles moving like this. Certain particles were like this in appearance, and they made a movement like this. So he drew like this. He drew in this way. And then he named this particle, sorry, he drew these particles and he represented these as his research work. And this was the first time some person took the hardship of drawing the diagrams of living cells. So Anton von Leeuwenhoek is accredited with the first visualization or first discovery of living cell. So who discovered cell? Robert Hooke. Who discovered living cell? Of course, Anton von Leeuwenhoek. Don't get confused, these two questions keep coming. Okay, now let us come to some other point. When we speak of cell, or or rather, when we speak of anything, suppose we speak of a building, we will say what qualifies this structure to be a building. Of course, this should be having a definite structure. This should have rooms, windows, doors, stairs if possible. There should be a well-built roof, and only then we can call this a building. But what qualifies a cell to be a cell? When we say cell, what is a cell? If we ask ourselves, what can you say? The first thing that comes to our mind is a sentence from the biology books. A cell is the structural and functional unit of life. But why so? Why do we call the cell structural and functional unit of life? Because just like while constructing a building, first you need a piece of brick. Other than or without the brick, you can't construct a building. Similarly, when we think of a living body, the smallest part of a living body which shows all its properties is a cell. That is why it is called the structural and functional unit. That cell, it possesses all the functions of the living body. Everything that a human being does or an animal does or any living thing does is done by all its cells. For example, respiration, yes. Nutrition, yes. Circulation, excretion, reproduction, response to stimulus, you name it and you have it, all the functions in that particular cell. But today we can say, I can tell you, I can discuss it with you, or you also might relate with me. That yes, this is something that we all read. But how did these things come to our discussion? Because some people again did a lot of work, they contributed their entire research life so that we could come up with something called cell theory. There are different forms of the cell theory, but today as we are discussing cell, let us not go into cell theory. In the next class, let us have a discussion on cell theory. Let us at the moment concentrate on cells. So, cells, broadly if we classify, there are two types. Living cells are classified into two broad categories. One, prokaryotic cell, the other one, eukaryotic cell. Let us write this down. So prokaryotic cell and eukaryotic cell, the two types, the two forms of living cells. Prokaryotic cells are quite primitive in nature. Eukaryotic cells are the more modern forms. Prokaryotic cells and eukaryotic cells, their difference comes as a point of importance in exam. Prokaryotic cells, they don't have a definite nucleus or cell organelle. Just like a bacteria, it has got an outer cell wall. Sometimes there is a slimy cover called glycocalyx or cell core. Let us draw another. Anyway, drawings always make things easy. So here is our bacterial cell. Our bacteria, it is having a long flagella. Many bacteria have got a flagella for moving. So outside the bacteria, there is not all bacteria causes this thing, but there is this cell coat in certain bacteria or glycocalyx, which is mostly present in bacteria which are present in extreme environmental conditions. So we are having a cell coat outside our bacteria, and inside the cell coat there is a cell wall. Inside the cell wall, there is of course the cell membrane here. flagella is connected inwards. So looking for it like this. So now inside the bacteria, there is a
nucleoid, which acts as the genetic material of the bacterium. Actually, bacterial cells do not contain any true nucleus.
oops, I didn't label. So let us label. This one is the cell core. This is the cell wall. Always remember that structures within a prokaryotic cell might in some cases be similar to eukaryotic cells, but
The internal biochemistry and structure might vary with variety of cells.
there. For example, the cell wall of bacteria contains more of peptidoglycan, but in plant cells also there is cell wall, but that cell wall contains more of cellulose. The constituents include a high concentration of cellulose in plant cell walls, but in bacteria it is peptidoglycan. Anyway, here we are having a nucleoid. The cell membrane over the level. Now there is one more thing to understand. Bacteria doesn't have any definite cell autonomy. How will the bacteria perform its vital functions then? This bacteria has developed, not this one alone, all bacteria have developed certain structures as per their necessities. So here there is a mesosome which looks very much like a twisted structure. Just like the sweet jalebi, it looks twisted like a jalebi. Jalebi, when you eat it, it has got an outer covering and inside you will find a fluid, which is actually sugar syrup. Inside the mesosome, this structure is called mesosome, which is a respiratory structure. Let me write it down here. So inside the mesosome, just like the sugar syrup inside the jalebi, inside the mesosome there are enzymes which carry out the respiratory functions. All the reactions of respiration. Of course, we will be having classes on respiration also. I'll be discussing how the chemical reaction of respiration keeps on going. But for the moment, let us just concentrate on the structure through which the prokaryotes respire, the mesosome. Inside the mesosome, enzymes are there for respiration, and those enzymes help the bacteria to respire. Now, let me tell you one thing. We keep on saying bacteria, but the term bacteria is not singular. There is no bacterias. Bacterium is the singular form. Bacteria is the plural form. So, this bacterium, not bacteria, but bacterium, has got mesosome for respiration. What if it is a photosynthetic one? What if it does photosynthesis? Bacteria, some of them are photosynthetic, some are non-photosynthetic. But what if this is a photosynthetic one? So let us think of how it will prepare its own food by photosynthesis. In that case, the cell membrane has got some structures in foldings. We call this, in terms of biology, we call this infoldings, that is inward foldings. And they contain photosynthetic pigments, especially bacteriochlorophyll. And these structures here, all blue, so I'm using a red color here. These structures are called thylakoids. These thylakoids help the bacterium to respire. And of course, at the end, for movement, it has got flagella. Some bacteria don't have a flagella. Instead, they have got a definite set of structures. These are many in number all through the body of the bacterium. And these are also locomotory structures. These are called cilia. In certain cases, you might find a term pilus. Pilus is something else. Pilus is a reproductive tube. It does not help in locomotion, but it helps in reproduction. Again, bacterial reproduction is something which holds good for another class. The first class today, we started discussing cell, we discussed prokaryotic cell. Eukaryotic cell, we will be discussing in the next class too. So, for our detailed discussion on eukaryotic cell, please tune in to our next class. But before we go, before I end the class, let me just tell you what is important from here. Nucleoid in the bacterium, this nucleoid is not the nucleus. It is a highly twisted and highly coiled DNA thread. It is the genome of the bacteria. It is the DNA of the bacteria. But the big deal is that unlike our DNA, which is thread-like in nature, the DNA of bacteria is actually a circular DNA. It is round. It is not
linear.
You can say it is a round web which is twisted and it is packaged inside the cytoplasm within the body of the bacteria. That is the nucleus. And from here, what are important things? From prokaryotic cells, from bacteria, what could be the possible questions? What is the main content of the cell wall? Peptidoglycan. So, bacterial cell wall. What is the main content? Let me write here. Bacterial cell wall. Main content is peptidoglycan. Next thing, genetic material, circular DNA. Okay, so this is the second thing. I hope everyone can see. So there are some other things, also some other points. So what else is for us to find from this? Yes, locomotion. Cilia and flagella. So what else could be right here? No complex cell organelles. So these are the basic things. I'm just talking about the basic points. Now this external cell core or glycocalyx, it is present only a few bacteria. Others, they don't have this.
While discussing bacteria, another important thing
It's called gram staining. What is gram staining also is one of the things that we are going to discuss in the upcoming classes. So this is the thing. One more thing I forgot to mention here is that about what I said, cell coat may or may not be present. So that's all about the structure. Prokaryotic cell. While discussing prokaryotic cell, we take bacteria as a standard. There are other prokaryotes also like mycoplasmas. So I hope this much was of use to you. I hope my discussion could bring some light on the topic of cell. In case you have any queries, you want to ask any questions, you are most welcome. Please do write in the comment section and let me know what your queries are, what do you want to know from our today's discussion. Also, let me know what more should I bring into my next discussion, what should be the topic of the next class. And we will be again joining in for another class on the next days, Friday. Next class will be a live one. We shall be live I shall go live. I shall be online on Sunday at 7 p.m. in the evening. Hope all of you will join in. And we will be continuing our discussion. We'll be having more of our discussions. And of course, there will be another such online video on Tuesday. So from now on, please log into the channel and please let me know where are you from, what are your interests in biology, and we'll be continuing with our classes the next week. Thank you.